So no doubt you have heard about these new variants of coronavirus 19 or COVID-19. And these new variants are very alarming. They are named for where they were first detected. So we have our UK variant, our South African variant, Brazilian variant, and they all have something in common. They all seem to be more transmissible. Well, it turns out that they all have something else in common as well. They all have experienced mutations, and not just mutations anywhere in the genetic code, but mutations specifically in a region that codes for this protein right here that's shown in red. That protein is known as a spike, and spikes are critical for a virus getting into our cells. So basically the way this works is the coronavirus has a bunch of spikes on the outside, and the spikes, being protrusions, need to bind to some receptor on our cell. And how infectious that virus is to our cells is somewhat reliant on how well they fit. If they only have kind of a poor fit, then it might take that COVID virus a while to get in there, and maybe it just falls off and it never ever binds. But if the spike is supremely designed, meaning it's got a 3D conformation that allows it to really tightly adhere and therefore get into our cells, then that's going to be a version that is much more infectious and therefore also easier to transmit. In other words, it's better adapted for humans. Now, unfortunately, the new variants that are causing alarm to scientists have not really been well studied yet. I should say the studies are probably ongoing. The studies have not been published yet. But we can turn to a variant that was established in early 2020. Now, this particular variant has a missense mutation that also affects the spike. And this missense mutation occurs at position 614 on the protein that codes for the spike. And it is a missense mutation that swaps out aspartic acid for glycine. Now remember, aspartic acid is electrically charged. Glycine is nonpolar, so this is actually a pretty big change. And it turns out that that missense mutation causes a drastic conformation in the spike. And so the spike ends up being kind of like folded over a little bit. And you almost think of it like if this is the head of the spike that is meant to bind to the receptor, think of having like aspartic acid right here, which is electrically charged, being attracted to something positively charged down here and kind of getting pulled down a little bit, so that it's really hard for that spike to really open up like a flower and bind. Well, if you change that piece that's kind of hooked down with that electrical interaction, and you change it to something nonpolar, nonpolar doesn't want to interact with anything positively charged. So guess what? It pops open. And that's exactly what we see. So the original conformation of that spike protein, which actually did not fit very well into our receptors because it was so closed, experienced a mutation, a missense mutation, where the aspartic acid got changed into glycine, and that caused all of these changes here. And so we can actually see that in this cute little GIF here. It opens up like a flower. There's the original mutation, now everything opens up. So the consequence of this is that it takes fewer viruses to infect us. And because the viruses are more able to bind to our cells and infect us, we end up creating a greater magnitude of viruses, meaning the viruses have an easier time even just jumping from cell to cell. And so consequently, we shed a lot more viruses as well. So we shed more viruses, it takes fewer viruses to infect another person. And you could see how this version over here would quickly become the dominant version. So how does that impact mortality rate? Well, interestingly enough, being more transmissible in and of itself leads to higher death counts. And here's why. This hypothetical scenario talks about an original virus that is not very transmissible, and then one that, let's just say it was a little bit more lethal, meaning that once it got into a person successfully, it increased the mortality rate, but it wasn't more transmissible. So you actually don't get that many more deaths, right? It's not very transmissible. hard to pass from person to person, like the original SARS was and the original MERS was. But now you've got a mutation such that allows this new version to transmit really, really easily. Well, let's say that the mortality rate is 2%. And so the mortality rate itself doesn't change. By sheer numbers, in other words, by simply infecting a whole lot more people, 2% of, let's say, 1,000 is going to be 20. But let's say now this new version infects 10,000. Well, 2% of 10,000 is going to be 200. So by itself, just being able to infect more people means that your death counts are going to be higher. And so this is one application of the consequence of missense mutation. And like I said previously, most missense mutations are bad and not helpful and often fatal. But occasionally a missense mutation arises that actually helps the organism. And here is a case where the mutation helped the virus. So it was good for the virus, bad for us. I have no doubt that the new variants that we see, the UK variant, the Brazilian variant, and the South African variants, are also going to show certain types of mutations in their spike protein that allow it to even more easily bind and get into our cells.